we can see that rods and cones are directly connected to the retinal pigment epithelium. Now, this epithelium has many physiological roles. Among the most important are to absorb excess light, to nourish the photoreceptors, and to help regenerate the rhodopsin. So then we have the photoreceptors. Remember, we have two types: the rods, which we mainly need in the dark, but also detection of movement, and then the cones for color perception, but also movement. So then we have the horizontal cells. Remember, we discussed in the previous part the lateral inhibition. We discussed that the horizontal cells inhibit a number of neighboring cells. Let's take a look at this again. Assume I would like to have a light focused on this one single cone. So the light is falling onto this whole region, but would like to focus it to contain it in this region here. We can see here that one horizontal cell is connected to a number of rods and cones. This one single horizontal cell can keep this, for example, just one photoreceptor active, while the others, the neighbors of this photoreceptor, are inhibited. The neurotransmitter used by the horizontal cells is believed to be glycine or GABA. Both neurotransmitters are always inhibitory in nature. So the activity of the horizontal cells is lateral inhibition to enhance the contrast. Then we have the bipolar cells. We discussed in the previous part, they convert the hyperpolarization in the phototransduction cascade into depolarization. Okay? Then we have the ganglion cells. This is where the first action potentials are generated, and the action potentials are carried away by the optic nerve. Then we have the amacrine cells, which I will discuss in the next slide. So we discussed the horizontal cells, now the amacrine cells. Their roles remain debatable. Here I have some notes here. The amacrine cells are the major carriers of rod signals to the ganglion cells in the retina. As such, they play a role in speeding up the slow potential rod messages for presentation to ganglion cells. Their distribution in the retina suggests that they tile the complete retina. Here you see one amacrine cell. The molar cells, in the blue asterisk here. You see they span the whole layer. I have the notes here summarizing. Molar cells function as optical fibers, directing and concentrating the yellow-green spectrum of light, to which many cones are tuned to respond maximally. Blue light seeps out of the molar cells to activate rods. Let's take a look at the fovea centralis and the distribution of the photoreceptors, cones, and rods. You see here the distribution of the cones in the fovea centralis. In the fovea centralis, we have the cones. We do have also some cones around the fovea centralis, but their numbers decrease dramatically as we move away from the fovea centralis. Then we have the cones, the rods here, you see, around the fovea centralis, all over the retina, we have the rods. We have here the so-called optic disc. Here, this is the blind spot. There are no photoreceptors here. This is the site where we have the optic nerve carrying away the signals, the action potentials from the ganglion cells. As we see here, the fovea centralis is a depression. This depression is in the center of the macula. Graded action potentials in the retina. Remember, we discussed that in the photoreceptors, we have graded potentials. In the case of phototransduction, the state of the photoreceptor in the light, we describe there that we have hyperpolarization. So the graded potential here, hyperpolarization. Then we have two types of bipolar cells, the on-center bipolar cells and the off-center bipolar cells. In the case of on-center bipolar cells, we convert the hyperpolarization into depolarization. Still, we have a graded potential here. Graded potential here, graded potential here. The first action potentials are generated in the ganglion cells and are carried away by the optic nerve to the brain. I will now discuss the physiological principle behind color perception. Before I get into the details, typically, the human eye can detect wavelength from around 380 to around 700 nanometers. Here we have photoreceptors and their light absorption. The cones, here, blue cone, green cone, red cone, and the dots. Here I have the notes for you. S for short, the blue cones, most sensitive around 430 nanometers. M, medium, green cones, most sensitive at around 530 nanometers. L, long, most sensitive at around 560 nanometers. In all cases, we have rhodopsin. However, there are differences in the sequence and the types of amino acids in the different rhodopsins, which means we have different absorption maxima. The key to color perception is to understand that they overlap. It is the degree of overlap, and where they overlap, that gives us different colors. Here we see an example. We have here the GIF, the three primary colors, and then we have here the absorption maximum and the overlap. Let's take an example here. Let's assume we shine a monochromatic light with a wavelength of exactly 580 nanometers. Please follow this dashed line. In this case, a monochromatic light with a wavelength of about 500 nanometers, it stimulates to 99% the red cones, and it also stimulates to 49% the green cones. This results in the perception of the color orange. Another example, a monochromatic light here, this dashed line, of say 440 nanometers, it stimulates the blue cones to 97% and hardly the green cones and not at all the red cones. We perceive this monochromatic light as blue. Here I have some more notes for you. I hope this was helpful.